Hi, this is Dr. Launen, and this is a lecture on the topic of RNA structure and function. The goals of this lecture are to learn the basic structure of an RNA nucleotide, to consider how internal base pairing, that should be internal, can introduce unique 3D structures to an RNA molecule, and to learn a few basics about the structure and function of the three major categories of RNA, M, T, and rRNA. And then we'll briefly address the topic of what is called small interfering RNA. So RNA is ribonucleic acid. That's what the RNA stands for. It is a member of the class or of macromolecules called nucleic acids. The other member, of course, being DNA, deoxyribonucleic acid. All RNA is made of 4 nucleotides: uracil, adenine, guanine, and thymine. The shorthand for that is U, A, G, and T. RNA serves several different purposes in the cell and is particularly important in the synthesis of polypeptides, which when folded become proteins. RNA is capable of base pairing just like DNA. So shown here are some DNA and some RNA nitrogenous bases here at the top, and those nitrogenous bases fit into the intact nucleotide of either DNA or RNA. So you should know a little bit about DNA nucleotides by now. DNA nucleotides look like this. They all have a 5-carbon sugar called deoxyribose, a phosphate group, and then a nitrogenous base, which could be T, A, C, or G. Any one of those, that's the nitrogenous base part. Here's an RNA nucleotide, and it looks incredibly similar. Same phosphate group, still have a 5-carbon sugar. It's just that the sugar here is called ribose, and the only difference is that it has a hydroxyl group at the number 2 carbon, whereas on deoxyribose, there's an hydrogen group on the number 2 carbon of this sugar. There's a little bit of differences in the base, the nitrogenous bases as well. Adenine, guanine, and cytosine for RNA are exactly as found in DNA. However, uracil replaces thymine. So we say in RNA there are no T's, there are U's instead. So here's an RNA nucleotide that has the nitrogenous base uracil. This is called uracil. So some other differences between DNA and RNA. Okay, we've already mentioned that RNA nucleotides contain the sugar ribose, whereas DNA contains the sugar deoxyribose. We've also already mentioned that RNA has uracil instead of the thymine found in DNA. Another difference is that RNA will be produced and then utilized as a single strand, not necessarily a helix, that's a bit of a typo there. It's a single strand, whereas DNA is always found in cells as the double helix, double-stranded DNA. So we'll think about RNA as single-stranded and DNA as double-stranded. All right, so here is a single strand of RNA. And here we've got the RNA nucleotide C, then A, then U, and then G. Okay? So when these are linked together, when RNA nucleotides are linked together through the process of RNA synthesis, another word for that is transcription, which is the same thing as RNA synthesis, those nucleotides are connected together by what are called phosphodiester linkages. So this is a phosphodiester linkage. It's not called a bond here because it describes this whole pattern of connectivity. Here's a phosphodiester linkage, here's a phosphodiester linkage, and here's a phosphodiester linkage. Okay? So that pattern is the same as observed for a single strand of DNA within a chromosome. You'll also note that there are ends to this molecule. So just like in DNA, there is a 5' end and there is a 3' end. In this figure, the 5' end is up here and the 3' end is down here. Most of my students remember that because they just memorize the fact that the phosphate group will be poking out on the 5' end and not poking out on the 3' end. The actual chemistry that explains that is requires an understanding of these sugars. So this little molecule here is a pentose sugar. It is the pentose sugar ribose because this is RNA. And it has carbons everywhere these lines connect. So there's a carbon, there's a carbon, there's a carbon, there's a carbon, and then this carbon is considered part of this sugar, okay? This is the number 1, 2, 3, 4, and 5 carbons. That was actually shown on this slide back here. 1, 2, 3, 4, and 5. If the 1, 2, 3 carbon is closest to the end, it's the 3' end. If the 1, 2, 3, 4, 5th carbon is closest to the end, then this makes this the 5' end. And that's true for DNA as well. RNA can base pair. So because we've got these nitrogenous bases, which love to form relationships with their partners, their base pair, that happens in DNA between complementary and antiparallel strands, but it also happens in RNA internally. So when the single strand of RNA is produced through transcription, also called RNA synthesis, if you have regions within the strand that are complementary, then this single strand can flop around until those nitrogenous bases come together, and hydrogen bonding called base pairing will occur. It's shown here as these little dashed lines. And the base pairing in RNA will be CG, just like in DNA, but here it will be AU, or the converse, UA, because there is no T in RNA. So the base pairing between RNA or within RNA molecules is CG and AU. This internal base pairing stabilizes strands of RNA and makes them more resistant to breakdown in the cell, and it also gives them a more complex 3D structure which can allow them to perform necessary functions in the cell. So shown here is the synthesis of RNA, and when RNA is synthesized, it's called transcription. And so what you get is this single strand of RNA. If it's messenger RNA that is produced, it will usually just stay as a floppy, like not internally base paired strand. If it's tRNA or rRNA, you'll get a lot of internal base pairing, and we'll look at that in a moment. A lot of the major categories of RNA are very important in determining the identity of proteins during the process known as translation, and we'll get to that later as well. So let's think about the different categories of RNA. Actually, let me go back and point out mRNA again. So the transcript, if the RNA is used to identify which amino acids are going to go into the peptide, then that RNA is called M for messenger RNA, because it's carrying the message about information to the structures that do translation, which are the ribosomes. Transfer RNA, also known as tRNA, is another category of RNA, and it's shown as like these little like blocky yellow structures, and each of them is composed of a single strand of RNA that's folded into what is actually not a square but kind of a cloverleaf structure. And on one end of it, each transfer RNA will carry an amino acid, and it will carry that amino acid into the ribosome, helping out with translation. That's the job of tRNA, to carry amino acids into the ribosome. And when we talk about translation, we'll talk about how the tRNA molecule makes sure that the information in the messenger RNA, which is shown here in teal, is used to get the correct amino acid. Okay, what that relationship is maintained by the tRNA through something called the anticodon region. For now, I want you to be aware that transfer RNA is a category of what is called functional RNA. It has a function in the cell that is to carry or transport amino acids into the ribosomes during translation. Those amino acids get put back onto, like new ones get put back onto the tRNA molecules by an enzyme that exists in cells called aminoacyl tRNA synthase. Here's what tRNA looks like, with a little bit more realism to it. So here is a single strand of RNA in a cloverleaf structure. It's got a 5' end on one side, and then if we follow it all the way around, it's got a 3' end on the other, and we looked at a picture illustrating that earlier. There are some funky sort of post-synthesis modifications done to tRNA molecules, so there's some interesting chemistry that's happening indicated by color that we're not going to get into. I really just want you to focus on the internal base pairing here and mention again that internal base pairing within transfer RNA allows it to take on a 3D structure that will let it carry amino acids, which will attach on this end, and then fit into the ribosome very precisely and actually attach to the messenger RNA within a ribosome, facilitating translation. Another interesting thing about transfer RNA molecules, if you look at the codon table that tells us what messenger RNA codons are going to be responsible for dictating what amino acids are chosen by the ribosome. If we look at that codon table, we'll see that there are 61 different, not amino acids, but codons. Okay. And yet there are only 40 transfer RNA molecules. So what we see is that each transfer RNA molecule that carries amino acids, here's a transfer RNA molecule that's carrying leucine, and here's another transfer RNA molecule that's carrying also leucine. If you look at the codon table, you'll notice that there are several different codons that encode leucine that can be found on the messenger RNA strand. They will typically only vary at the 3rd position. So what happens is these two are actually the same tRNA molecule, but they're able to slot into the ribosome whether you've got CUC sitting there or CUU sitting there. So they're going to slot in and they're going to carry leucine in, and they'll be pretty tolerant about some mismatching at the 3rd position that is uniting this messenger RNA codon with this tRNA anticodon region, the spot that's responsible for ensuring the right tRNA gets into the ribosome structure during translation.